Partial linkage is explained by the behavior of chromosomes during meiosis. Thomas Hunt Morgan made the conceptual leap between partial linkage and the behavior of chromosomes when the nucleus of a cell divides. There are two types of nuclear division, mitosis and meiosis. In mitosis, the diploid nucleus of a somatic cell divides to produce two daughter nuclei, both of which are diploid. During a human lifetime, approximately 10 raised to 17 mitoses are needed. Before mitosis begins, each chromosome in the nucleus is replicated, but the resulting daughter chromosomes do not immediately break away from one another. To begin with, they remain attached at their centromeres and by cohescent proteins which acts as molecular glue holding together the arms of the replicated chromosomes. The daughter cells do not separate until later in mitosis, when the chromosomes are distributed between the two new nuclei and each of the new nuclei receives a complete set of chromosomes. During interphase, the chromosomes are in their extended form. At the prophase, each chromosome has already undergone DNA replication, but the two daughter chromosomes are held together by the centromere. During metaphase, the nuclear membrane breaks down and the chromosomes line up in the center of the cell. Microtubules now draw the daughter chromosome towards either end of the cell. In telophase, nuclear membranes reform around each collection of daughter chromosome. The result is that the parent nucleus has given rise to two identical daughter nuclei. Mitosis illustrates the basic events occurring during nuclear division, but is not directly relevant to genetic mapping. Meiosis occurs only in reproductive cells and results in a diploid cell giving rise to 4 haploid gametes, each of which can subsequently fuse with a gamete of the opposite sex during sexual reproduction. The important distinction is meiosis involves two nuclear division, one after the other, whereas mitosis is just a single nuclear division. During mitosis, homologous chromosomes remain separate from one another, each member of the pair replicating and being passed to a daughter nucleus independently of its homologue. In meiosis, however, the pairs of homologous chromosomes are by no means independent. During meiosis 1, each chromosome lines up with its homologue to form a bivalent. This occurs after each chromosome has replicated, but before the replicated structures split, so the bivalent in fact contains 4 chromosome copies, each of which is destined to find its way into one of the 4 gametes that will be produced at the end of the meiosis. Within the bivalent, the chromosome arms or the chromatids can undergo physical breakage and exchange of segments of DNA. The process is called crossing over or recombination and was discovered by Janssens in 1909. How did the discovery of crossing over help Morgan explain partial linkage? To understand this, we need to think about the effect that crossing over can have on the inheritance of genes. Let us consider two genes, each of which has 2 alleles. We will call the first gene capital A and its alleles capital A and small a, and the second gene B with the alleles capital B and small b. Imagine that the 2 genes are located on chromosome number 2 of Drosophila melanogaster, the species of fruit fly studied by Morgan. We are going to follow the meiosis of a diploid nucleus in which one copy of chromosome 2 has alleles capital A and capital B and the second has a small a and small b. Consider the 2 alternative scenarios.